Morning. My name is Tom Lynch. I'm here from Aldevron, here to provide an overview of some of the services and products that we've developed over the years in support of cell and gene therapies. Also present a few highlights of some work that we've done in collaboration with our partners and clients. In celebration of our 25th anniversary last year, the theme of that celebration was about advancing cell and gene therapies, advancing every day. On the right, we highlight some of the milestones throughout Aldevron's history, the development of GMP source quality system, first plasmid GMP released, first off-the-shelf Cas nucleases released as GMP quality. Throughout all of the tens of, now over 165,000 lots of plasmid DNA released, to more recently the investment that we've made in support of mRNA as a modality. So as you kind of look at how we're advancing our capabilities, our convenience to our clients and partners, our facilities, and the efficiencies that we can help our partners and clients gain throughout their journey to clinical and commercial applications. Aldevron primarily focuses in three areas of manufacturing. One is plasmid DNA. This is the start of Aldevron, doing custom manufacturing for our clients and partners for viral vector plasmids, as well as linear templates for mRNA manufacturing. We also produce proteins. More recently, it's been keenly focused on our enzymes in support of mRNA manufacturing, as well as gene editing nucleases, but we also do produce custom materials for therapeutic and diagnostic applications. And then messenger RNA. This has been work that we've done for several years in producing research use material, but also now through drug product. And so when you look at Aldevron as the manufacturer, 2.5 years ago, we were acquired by Danaher, who recently rebranded as Danaher Life Sciences. And shown on the right are several of the opcos within Danaher Life Sciences. So think about kind of the collection of the organizations and the services, products that we supply. We're really starting to create, more value work streams for our clients and partners through our capabilities. It's been quite an exciting past couple of years. So I just want to kind of start with the basis of Aldevron, and that's our custom manufacturing. Plasmid DNA, we produce traditional supercoiled and linear templates. We also have brought on new technologies, next generation plasmids with our nanoplasmid vectors. And we've also continuously, over the last 25 years, worked to improve our upstream and downstream processes to manufacture. On RNA, we're producing our own linear template. We're producing our own IVT and capping enzymes for production of mRNA, and we produce this material from research use. And then on the protein side, again, custom manufacturing, I would say more recently, primarily in support of cell and gene therapy, gene editing applications. But we do produce material that also deliver into diagnostics applications. When we look at the facilities, our footprint is primarily in the Midwest in the United States. Our headquarters are in Fargo, North Dakota. It's usually sunny and warm there. But we have purpose-built facilities in support of research use in clinical material and commercial applications. In Madison, Wisconsin, this is where our protein group is located, as well as our innovation lab for furthering process development and testing applications. And then our most recent facility in Lincoln, Nebraska is where our nanoplasmid technology, as well as then the continued improvement within the cell line and the feed to grow those cell lines. So we've made very deliberate choices and built purpose-built facilities to support cell and gene applications. And a little bit more in the details, in our breakthrough campus, this is where our clinical manufacturing occurs across approximately 300,000 square feet of space. We have over 40 suites for production. We're generally functioning at 30 and 300 liter scales, can also do shake flask. As far as the research grade in our advanced campus, we were producing, tens of thousands of plasmid lots per year in approximately 60,000 square feet. The scale, again, is, shake flask to approximately 30 liters. On the protein side, we can go to a little bit bigger scale. We do have stainless fermentation there. We can go up to 1,000 liters for your protein requirements, and that facility is ISO 1345 certified. And then our Impact Campus, which again is our Center for our Nanoplasmid Technology. For all of these applications, our primary host for expression and production is E. coli. We do some mammalian work, but nothing to support GMP there. And this has all been built, as we kind of see the advancement and the expansion of cell therapies. Also, we're keeping a keen and close eye on, the modalities that are required for new and emerging therapeutics, as well as how they are delivered. When we go beyond, we think about, custom manufacturing and what's next to drive efficiencies for our clients and partners. We have built a small but very focused catalog of off-the-shelf products. This allows our clients and partners to take small quantities to do proof of concept work, maybe development work. And we offer this off-the-shelf material for viral vector plasmids as well as Cas nucleases and IVT enzymes from research use through GMP. So therefore, when you're thinking about, development of your clinical program, you can test a small quantity of GMP grade material without having to, do a single batch of large quantities. And then when we think about the custom services and the off-the-shelf products, we start building workflows, sequence to vial for mRNA. We're also working with our sister opcos in that regard, as well as in the genomic medicine space with IDT, when they're producing guide RNAs from research use to GMP, combining that with our custom ability to manufacture nucleases and off-the-shelf nucleases to combine those into ribonucleoprotein complexes or RNPs. I'm just going to highlight a few products and services which in the three areas in which we primarily operate. For a viral vector space, we have for several years now supported off-the-shelf reagents of helper plasmids, rep caps, as well as backbones for transgene expression for AAV manufacturing. These helper plasmids do come along with a DMF support. So these are materials off-the-shelf research use through GMP. For the lenti space, again, all that's required for your packaging and transgene requirements to produce lentivirus. The true benefit of these two sets of off-the-shelf products are that when it comes time to manufacture, 2 or 3 of the plasmas are already produced. They're waiting for you. You don't have the time or the investment to make for a custom batch. We only need to focus on the transgene expansion and purification. And then finally, in the DNA area, our nanoplasmid technology. If you're not familiar with those, these are reduced size plasmids, small backbones, also eliminate the need for antibiotic resistance marker, been shown to improve transgene expression and also decrease costs for our clients and partners. Can also be used as an HDR template for gene editing applications. So lots of use within transgene cargo delivery as well as on the gene editing side. Messenger RNA. This is an area that we got into in 2017 with our first clinical batch of material delivered in 2018. But through our partnership of our sister opcos within Danaher Life Sciences, under the umbrella of Cytiva with Precision Nanosystem and their LNP technology, and VanRx with their fill finish capabilities. We've built a work stream to support from electronic sequence to vial delivery to our clients for drug product. The benefits here is that we're producing the DNA linear template. We're also producing the enzymes to not only manufacture the transcript, but also be able to do enzymatic capping if that's the direction that our clients and partners decide to go. So this is, a lot of experience and expertise and support all under one roof. Finally, with the proteins, I'm just going to highlight, again, work that we're doing in coordination with IDT on the guide RNA side, but this is working to streamline RNP production. We spent a considerable amount of time and resources developing the test panel for release of clinical RNPs. This is work that we've done for a large number of clients and partners. And the benefit here is obviously the test panel, but also you have the opportunity to come in and have us produce your nuclease of choice on a custom basis to make sure that we are further optimized for the RNP manufacturing process, but also you have the option to take any number of our off-the-shelf enzymes from our wild-type SP-Cas9 to our high-fidelity Cas9 SPI-Fi, and more recently through our licensed type 5 nuclease MAD7, which we have since rebranded as Eureka 5, which has been kind of popular in the allogeneic space. And when you kind of take, all the work that we've done to advance our custom services and our products, I just want to highlight, a few areas of work that we've done in coordination with our partners. So this is some work done by Abby Stahl and Jennifer Doudna at the IGI in Berkeley. The requirement here was they needed a very low endotoxin sample of Staph aureus Cas9 in order to in vivo delivery into mouse brain. And we were able to accomplish ultra low endotoxin, levels below 0.15 EUs per milligram of protein. And so they did a comparison of the RNPs compared to the Staph aureus Cas9 packaged in AAV, and found that the RNPs had higher editing efficiency, and obviously the prep had much lower endo levels than a standard prep. Kind of along the same line, Christian Capitini and Chris Saha at the University of Wisconsin working to work on a vector or viral-free CAR T cell line, and we had produced not only the Cas9 but also the RNPs in order to do a knock-in of a CAR. They tested this against a retrovirus CAR, and it led to a better anti-tumor response with a less exhaustive phenotype. Kind of along the same lines, but Stanford and Aldevron did work in coordination with HDR template delivery with nanoplasmids. So using Aldevron produced RNPs and nanoplasmid as the HDR template, they were able to show that they were able to knock in a CAR. Using nanoplasmid, they did this in comparison with retroviral CAR and saw, similar benefits, but kind of the main takeaway that they found was that there was some real cost efficiencies gained using nanoplasmid as a HDR template compared to linearized DNA. And then finally, work with Roche Genentech. Again, this was using nanoplasmid as an HDR template, but this was using RNPs generated with our SPI-Fi nuclease. And they found that comparing the knock-in efficiency compared to linearized DNA as well as POC57 carrying the cargo, that they were able to achieve increased knock-in efficiency and cell viability. So we kind of take a step back. and I'm probably going to go 1 minute over, so I appreciate your time. For the past 25 years, we've really been keenly focused on supporting and advancing the cell and gene therapy market. I think our experience is proven in terms of the numbers of successful batches that we've released for research, clinical, and commercial applications. We're also, looking to advance any technology within Aldevron, and it's exciting to see kind of new modalities and new deliveries coming about. When we take a step back and think about how Danaher is working to support, genomic medicines, we think about the sequence generation, amplification, the delivery, the manufacturing, the analytics, and you can kind of start to see how we're working to thread the needle between each OpCo and build really this great value work stream to advance the field. And another area in which we're working to support the field, this is Jennifer Dowden and Fyodor Urnov at the IGI in Berkeley. It was announced in January that Danaher has made an announcement, pictured here along with Danaher's CEO, Rainer Blair. It's called the Beacon Initiative, and the focus is supporting their efforts for 4 years so that we can develop gene editing therapies for 2 rare genetic diseases. So very proud of that work. And I just want to say thank you, everyone, for attending. Happy to answer any questions. During this break time, we have a table out in the general area. So thank you, and hope everyone has a great day.